Hello everybody, here's Andreas from Azure Biosystems. Today I would like to talk about our brand new Azure Sapphire Biomolecular Imager. So the Azure Sapphire is a scanner which can be used for different types of applications, like standard Western blots or gels, but the machine can do much more. For instance, you could put on the glass surface tissue and scan it in super high resolution, or we can even put full-size animals on the glass surface, like a mouse. And the webinar today is exactly about this topic. I would like to show you these additional applications which are possible on our device. Okay guys, so let's get started. At the beginning, few brief introduction words on our company. So Azure Biosystems, we are a manufacturer from US. We are designing, developing and manufacturing our products quite close to San Francisco and the Silicon Valley. And we have a team with over 25 years experience in research and development. The brand itself, Azure Biosystems, we placed in 2013 on the market. So we are still a young company, but what you can see here, we have already over 12,000 users globally. We have over 3,000 imaging systems in the world in the field. And when you look at the amount of publications, also this is rising exponentially. So the Azure Sapphire Biomolecular Imager will give you as a user more resolution, more sensitivity, more applications and more value. And here, this part is important for us. So the application part for us is important to give you the flexibility that you can do much more with this device. And to make this possible, we put inside different laser light sources. So we have installed RGB lasers into the scanner. We have inside also near-infrared lasers. In addition, we put inside phosphor imaging module and a chemiluminescence module. And here you can see which fluorescence laser light sources are installed. So for RGB, we put inside a blue, green and red laser. And for near-infrared, we are using these two types of lasers. So at the end, you as a user will have the choice which configuration of lasers you would like to have in your machine. When you are coming more from the RGB world, then it makes perfectly sense for you to use as a red laser the 658 nanometer laser. If you are coming more from the Western blot world, when you have done before a lot of near-infrared Western blotting, then we have an option for you. We can put inside a 685 nanometer laser. So you see, we are very flexible here. So what makes the Sapphire so unique is that we put inside three types of detectors. We have inside the PMT, the Photomultiplier Tube. We have inside the APD, the Avalanche Photodiode. And we have inside the CCD chip for chemiluminescence. And this combination of detectors you will only get from Azure. So we have also a patent in US on this technology that you can only get a scanner with these three types of detectors from Azure. And soon we will have this also the protection worldwide. So why are we using a photomultiplier tube? We are using the photomultiplier tube for blue fluorescence and phosphor imaging. And here you can see the reason why. So the photomultiplier tube has a higher quantum efficiency in the shorter wavelengths, as you can see here. On the other hand, we are using the Avalanche Photodiode for red, green and near-infrared fluorescence. And you can see here that the Avalanche Photodiode has a higher quantum efficiency in the longer wavelengths. Means the other way around. So you see, it makes perfectly sense to put inside these two types of detectors, the Photomultiplier Tube and the Avalanche Photodiode. Then you have the full range of quantum efficiency and you are very flexible on your applications. In addition, we put inside a true CCD chip. So in the scanner there is true CCD camera installed, and we are using here the CCD chip for the chemiluminescence detection. Well, for the chemiluminescence detection, you know that your bands are glowing up by the ACL reagents, and we need to collect the light, right? So this is why we have inside the CCD chip, which will collect the light which is coming from your bands, from the ACL reaction. For chemiluminescence, we have here also a nice feature. So when you have your Chemi Western blot and you have on your membrane color marker, we are able to capture this. Yeah, we are taking a color photo and we are pasting in then this image into the chemiluminescence image. So you have your marker on the right position, pixel by pixel on the right spot, which is a great feature. The field of view of our Sapphire scanner is for fluorescence 25 by 25 centimeters. And well, for the most types of applications, fluorescence applications in the lab, this is more than enough. For chemiluminescence, the camera is built in below the glass surface, so the field of view for Chemi is 16 by 13 centimeters. Also, this is more than enough for Chemi membranes, from when we have a Chemi Western blot. We have also a super high resolution. The Sapphire scanner has a resolution from 1000 micron to 10. And important to know is here we have true 10 micrometer resolution, so there's no interpolation in our machine. Also, the scanning speed is super fast. So our scanner is working with 50 centimeters per second with all 4 lasers simultaneously. And this is an important part. So this is very special. We can scan all 4 lasers. We can use them simultaneously. This will give you a really boost on speed. You don't need to wait anymore until one scan is finished and another laser is starting. We scan this in parallel. In the package is also included a computer. You can choose between a laptop or a high-performance desktop computer, which will come also with a big screen. Of course, the capture software is also included. And the capture software is easy to use. 5 minutes in front of the machine and you will know everything. So this is how it should be. You as a user should come to the machine and be able to scan very quickly. So we try to keep it very simple, to the point. So you just put your specimen on the glass surface, you select the area of view you are interested in, you choose the method, you choose the pixel size, the scan speed, the sample type, and you run your final scan. That's it, nothing more. The machine will generate for you uncompressed TIFF files, which is very flexible format. So you can put the TIFF files and put it in software like ImageJ, but we have also an own analysis software. Here's important to know that this is an option for you. Yeah, so we are not forcing you to use our analysis software. It's an option, right? So when you like to put the files into ImageJ, you can go ahead. But this Azure Spot is very professional. It has included automatic detection of bands. It can give you a report. We have different normalization, quantifications, insight methods. We have also analysis toolbox. Here you can select your bands manually and go ahead with the normalization and the quantification. We have also an array analysis inside and a colony counting module. So it's a very professional analysis software with a very dense, bright package of applications, of possibilities to analyze your data. But again, it's an option. Let's look at the configurations of our scanner. Well, so we are, as said, already very flexible here. You can choose as a user if you would like to have a Sapphire RGB only version. If you are coming just from the Western blot part, you can have the Sapphire near-infrared only or the Sapphire RGB near-infrared. In addition, we have a chemiluminescence module, so the chemiluminescence camera, the Chemi camera we can build in. We can have also as an option the Phosphor Imaging module. And the third option is the Q module. So what is the Q module? This is something new. Well, it's designed for people who are doing a lot of Western blots. So maybe you heard already there's a trend in the market. People are talking about Western blot 2.0. So what is Western blot 2.0? Maybe you realize already that people are starting to go away from the normalization from GapTH and beta-actin, the standard housekeepers. Why? Because over here it depends on the cell cycle a bit. The protein expression is going a bit up and down, right? So when you have a complex protein mixture, let's say you have inside 5,000 proteins or even more, it's a good idea to do the normalization with all of these proteins. So Western blot 2.0 is about total protein normalization. And you could take the Sapphire near-infrared and include the Q module. And the Q module is a green laser. And we are using the green laser for total protein normalization. We have also old stains, which are excited by our green laser, and the benefit is here you could use the 700 and 800 channels from the near-infrared for your protein of interest, and for the total protein normalization you are using the green laser, which is far away from the near-infrared, right? So there's no crosstalk. That's a big benefit here. And now I would like to talk about the additional possibilities of applications which are possible on our scanner. And as an example, I would like to talk today about mouse imaging samples. So all the samples you will see we have done together with our customers. Let's start with this here. It's a mouse heart. So the client puts the mouse heart on the glass surface, and before this they used iDISCO. So iDISCO is a protocol to get rid of the fat of the mouse heart. And when you get rid of the fat, then you can imagine the tissue is clear like glass, and the lasers are going really inside, very deep. So we have done this. You see here the glass, the mouse heart on the glass surface. Below the glass surface here you see our scanning head. And we moved also the scanning head a bit up. So this is also one of the additional advantages of our scanner. We can move the scanning head up and down in Z position, so you can do also kind of Z stacking. So in this case, we moved the scanning head up, I think it was around about 3 millimeters. And here I show you just a final image. So this is a merge of the 700 and 800 channel scanned with 10 micron, and the scanning time was here done in 8 minutes. This is an additional example. We used here cubic. So cubic is a protocol also to clear the tissue. And we cleared here E16 embryo mouse skull and scanned it with 10 micron. You see here the embryo mouse skull lying on the glass surface. And when I zoom in, you can see it here. By the way, so when I put tissue on the glass surface, I use water and put the tissue on this water drop. It's a nice lens effect, right? You get the images sharper because the laser is going better through water than through air, of course. Yeah? And in this case, interesting for us was the GFP fusion protein, so the signal which is here in the brain of the animal. Yeah, and this of course is not lying on the glass surface, so we had to move up the scanning head up in that position. I think here in this case we use 4 millimeters, move the scanning head up to get the image sharp, and I show you here the results. Let's go to the final merge. Here you can see it. So here's the GFP signal in the brain in green. Here we see also the eye of the animal. Well, this function by moving the scanning head up and down is very interesting. Yeah, we call it Z stacking. We are working also on it that we have this automatic, that we will push a button and the machine will generate automatic Z stacks. But also what is interesting to know, the 800 laser, the near-infrared laser, has the possibility to penetrate this tissue. So you don't even need to clarify it. And when you combine tissue which is not clarified, and we use the 800 nanometer laser in parallel with for instance the 488, when you combine 800 channel and 488, then you get nice overlays. Look at this. So this is an image which shows you the signal from the inside. This the 800 channel signal, yeah, which is penetrating the tissue, and the red signal, this is the 488 laser, which is of course just hitting here the surface. So you can imagine you can do this nice overlays to look inside, and you see also the structure in the front. Now I would like to show you some images from mouse embryos. So we have also clients which are cutting mouse embryos into slices. They put them on the microscopic slide and normally go to the microscope. But you can imagine a microscope as a very high magnitude, right? So even when you use a 5-fold lens, it might be a too strong magnitude to see the full specimen. So you can imagine a scanner can here do a very nice job. So let's look at this. Look at this. This is gorgeous. We see here the embryo on the microscopic slide. And we see very sharp here all the organs, which is great. We see the brain. Look at this. We can even see here the backbone or the fat. Look in the neck of the animal. Here we can see the fat. So very nice. Now I would like to show an additional mouse applications. What about a full-size mouse? To put a full-size mouse on the scanner. Is it possible? Yes, of course. Look at this. We put a full-size mouse on the scanner. In this case, we opened the belly, you see. And when I zoom in, we can see it very detailed, very sharp. You see all the hair. Now, and when we even zoom in more in the belly, look at you see here the veins and even the muscle fibers, very sharp. You see here the hand of the animal. So we had an experiment. Client had 3 types of mouse. They're using a wild-type mouse, a transgenic mouse, and the gene-sat mouse. And normally they were distinguishing between these 3 types of mouse by taking out the brain. So there's in the wild-type mouse low GFP expression in the brain, nearly nothing. It's a wild-type mouse, no GFP. In the transgenic mouse, a bit more GFP, and the gene-sat is the overexpression mouse, so a lot of GFP expression in the brain. And the question was, can we distinguish between these 3 types of animals, when we scan them in full size? And here I will show you the results. So we put the mouse on the scanner. Also here they are lying on this water drop. I use this water drop effect, as I told you before, and I close the lid. And no worry here, we have enough space, so this round about 3 cm space. So when we close the lid, the animals are not harmed. In this case, we are talking about here post-mortem animals, so they were dead already. And look at this. Here are the results. So wild-type mouse, transgenic, and gene-sat. And when we look at the mouth part of the animals, we can clearly see that the wild type has no GFP, transgenic more, and the gene-sat the overexpression a lot. Also the hand, when we look at the hand and the feet, there's a difference, right? So we can clearly distinguish between these 3 types of animals by scanning them in full size on our scanner. I can show you this also in black and white. So in black and white we see even the contrast better, so we can distinguish even better. So you see here no GFP, a bit GFP, and a lot. And of course we have been interested, is there a correlation to the brain, right? So we have taken out the brains of exactly these animals to check this. And look at this. So here's the wild type brain, the transgenic, and the gene-sat. And let's look at the GFP intensities. And you can see here, in the wild type brain, no GFP, a bit GFP, and a lot of GFP. So there's a clear correlation to the full-size mouse scans. Now I would like to show you some images of mouse brains scanned with 10 micron. So here you see a mouse brain scanned on our scanner. Here you can see the cerebellum. And as I told you at the beginning, we can do a kind of Z stacking also with our unit. So we can move the scanning head in that position up and down. And here I put the mouse brain on the glass surface and move the scanning head up. You see? And when you end up with Z stacking images like this, we can even put them together to a little video. I can show you this here, right? So this was a pre-scan Z stacking, and I put this together to a little video. So this is a nice function, and we can, right now we are doing this manually, but of course we can show you how we can do this. Another topic, what about mouse brain slices? So we have also a lot of clients who are cutting the mouse brains on clear state, right? And have the sequential cuts on the microscopic slide. So here you see how it looks like on our scanner. And this is in the fluorescence and color. And when we zoom in, It's great. Look at this. You can here see nicely also hippocampus, cortical parts, right? We have done also an experiment with a virus-injected mouse brain to localize the hippocampus. So you see the original slide, the microscopic slide. We put it on our scanner and scanned. And here are the results. And you can imagine also that you can use the full size of our glass surface. So you can even put more than one microscopic slide on the glass surface. And this is a big benefit, by the way. First, you save time, and second, you end up just with one image. Yeah? So when you move the slider back and forward, all of your images will change at the same time. Yeah? So it is a big benefit for comparison reasons. And when I zoom in, so you can see here the hippocampus. Yeah, in green even better. And we use also methylene blue injection into the mouse hippocampus. Yeah, to localize it. This is scanned with 10 micron. And here you can see nicely the methylene blue hippocampus. It is shown in red. What about mouse brains cut in a sagittal cut? So a full-size mouse brain cut sagittal. We put it on the scanner. Also here you see I'm using water for this water drop effect. And there was also a GFP fusion protein expressed mainly here in the cerebellum. So I can show you here this also in a different channel. And finally, the fluorescence color. So green is showing you the GFP expression, and in this case, very strong signal expressed in the cerebellum. Look at this, it looks very nice, very sharp. This is a mouse brain's epithelium scan. And we had also clients who put different body parts on the glass surface, yeah, to see if there is any GFP expression on any mouse, on any body parts. And here's the result. So it's in black and white. We can also show you this in the color. And when we zoom in, so here's the mouse brain. And here you can even see the bone, right? This is mouse liver. You can see nicely here the structures inside of the mouse liver. This is in a different channel. We can also zoom in and look at this. This looks great. It's mouse liver, very sharp, very detailed, and you see all the structures. And in this case, there was also GFP fusion protein expressed here in this liver structures. You see here in green. This is the second mouse liver I would like to show you quickly. Two different channels. And here are some additional tissue parts we put on the scanner surface, in this case from the inner part of the body, yeah, to look if there's any GFP expression. And you can see here that we have a strong signal in the heart, right? In the heart, also in the kidney. You see here the kidney below, yeah? And this one here is the liver. We have also clients who are interested in the vascular system. So they are working with mouse aorta. So you know aorta from the mouse, which is the biggest artery in the body of the mouse. And we put this on the scanner. I can even show another sample where we zoomed in a bit, and you can see clearly here also the aorta structures. It's a very tiny structure, by the way. Right. Now I would like to show you some mouse fat. So this is mouse fat on the glass surface. And here you can see a mouse hand bone. So we had also an experiment. Mouse hand, a healthy one and a disease one on the right side. I can show you this also in fluorescence, in color. And the nice thing here is that, I mean, it looks like an X-ray, right? But of course, it's not. In X-rays, the scanner has the ability with the 800 channel, the near-infrared channel, to penetrate the tissue. Yeah? And this is very nice. So we use here the 800 near-infrared channel to look also inside of the hand. And left, we can see the healthy one and the disease one on the right side. Alright, so I hope you like these examples of additional applications. Mouse imaging is of course a big topic, but of course we can do much more. If you want to learn more about the Azure Biomolecular Imager, I recommend that you go to YouTube. We have a nice video there. So just type in Azure Biosystems Sapphire Biomolecular Imager and you can see our video. Also, I recommend to you visit our website. Or when you have additional questions, please don't hesitate and write us an email to info@azurebiosystems.com. And of course, we would be very happy to meet you at your lab, to do some demo with you, to scan your samples on our scanner. So I'm looking forward to see you soon in your lab and test with you together our scanner. Thank you for attending this webinar. Take care and bye-bye.